Okay, I guess I can already start introducing the next speaker. Unfortunately, Dr. Silvia Blemker wasn't able to join in person. She's all the way in Virginia in the US, from the University of Virginia. And Silvia is a distinguished professor of biomedical engineering at the University of Virginia. She leads the Multi-Scale Muscle Mechanophysiology lab, which develops multi-scale computational and experimental techniques to study skeletal muscle biomechanics and physiology. She's also a co-founder of Springbok, which is a company commercializing image-based muscle analytics AI technology that was a spinoff from the research in the lab. And she's also a really great mentor I was in her lab at University of Virginia for my bachelor's and master's. So I'm really excited that she was able to join today in Barcelona, well, in Zoom over Barcelona. But Silvia, whenever you're ready, you can go ahead and share your screen and start presenting. I think we can't hear you right now.
Here. Can't hear. Can you hear me now?
Yes, now we can hear you.
Okay. Okay. Sorry. Oops. There we go. Back. Okay. All right. Should I start? Sorry. Hello?
Is there a way to make the window a bit larger? I'm not sure. Or I think this is, okay. Or Silvia, yeah, not on you, I don't think, but on.
The video is smaller and it's not from the person.
Yeah, I think this is from the back from the auditorium. Sorry, Silvia, 1 second.
No worries.
Okay. Yeah, I think right now we're seeing like a split screen of like half your face, half the presentation. So we're trying to fix it.
Yeah, you don't need to see my face. That's better.
That's much better, yeah. Okay, I think this is fixed, so you can go ahead and start now.
Awesome, cool. Well, thank you. Thanks for that introduction, Katherine, and for the invitation to be here. It's exciting. Sorry, I can't be there in person, but excited to share with you our work. I titled my talk "Same but Different," looking at how variability in muscle structure influences function using models and experiments. I've gotten, now that we have more technologies to quantify structure of tissues, and for me, I'm mostly interested in muscle. It's opened a window for us to start to really explore the relationship between variability in structure and function. I think we've always talked about structure and function of how of tissues more kind of from the higher level of like, okay, this tissue looks like this, that tissue looks like that. It's how it influences their structure. But now we can start to look at variability across a population, which I think is exciting and a direction I think would be exciting for the field. So I wanted to talk a little bit about that today. But first wanted to acknowledge my lab, which, the work that I'll be describing is the work of the students. We have Katherine here in a few pictures, even from her undergraduate days. So excited to share with you the work that and the ideas and the enthusiasm that comes out of all of the students in my lab at the University of Virginia. So there's many reasons why the students and I and my colleagues are excited about muscle in particular. One is that they're motors for all human movement, and there are lots of them. So, some muscles we think about a lot, some muscles we think about less, but we're interested in all of them, very inclusive in terms of all the different muscles. And there are lots of them, but they're all made up of the same elements. So if you zoom in real close, thinking multi-scale at the cell and subcellular level, we realize that they're all made up of the same thing, but the way that they're structured or arranged really influences their function in a really powerful way, such that they can be the same thing, can do all these vastly different functions. And then the other one that I was referring to at the beginning of this talk is the idea that mechanics varies widely across the population, reflecting the nature of diversity in muscle, and which could be a property of nature and nurture, given the fact that muscles are so highly plastic. Some genetic variation of how muscles are structured, but also the way we use them influences that a lot. So that for that reason, we see a lot of variability. So muscle structure and motor function is determined by structural variations at many length scales. And I think this is particular interest to this audience, all the way from whole body mechanics, all the down to the specific muscle, how it's organized, its architecture, to the composition of the tissue, down to cellular biochemical factors, which could vary. All of these influence the way muscles operate and the way we move. So the theme of my lab is to create computational models, and we're actually traversing all these scales of a wide variety of projects down to from the cellular level and tissue level, examining how muscle cells respond to stimuli and how that results in the tissue remodeling that we see in muscle over time. It's something that Katherine worked on for her during her graduate master's work in my lab in collaboration with Shane Pierce-Kotler, who I believe is giving a talk later today. And then up to micromechanics, looking at how muscle cells and ECM interact, and how the arrangement of these elements influence the mechanics to whole muscle level, all the way up to looking at dynamic models of movement. So we're interested in crossing those scales. I think it's a little bit mind-blowing, but I think it's powerful and important because you can't totally ignore the others, or you can sort of go you kind of lose sight of the purpose. Okay, so today, though, that's a lot of different things. I wanted to share a few examples from a range of muscles. So I did want to sort of highlight this like lots of different muscles concept, because I think it's such a cool thing about muscle that there's so many different, essentially different functions, and understanding that, I think, is a powerful way to really understand muscle mechanics. So I wanted to start with talking about speech, actually, and muscles involved in speech. So we've been interested in the muscles of the soft palate for some time. So if you think about the anatomy of your mouth, if you imagine looking into the roof of your mouth and seeing what's up there, you have a hard palate, which is in the middle there is bone. And then behind that, there's a soft palate. So okay, so you have the soft palate here. And so if you look, so the hard palate part is bone with mucosa around it, but in the soft palate, there's a group of muscles that are important for both speech and swallowing and breathing. And so if we look at, so the image on the left is showing what this looks like if you look within, up to the roof of the mouth. And then on the right here is a sagittal view, so seeing the structure of these tissues from that view. So if you look at this, there's one particular muscle called the levator veli palatini muscle. Oh shoot, did I get lost again? Hello?
We can still hear you fine.
Oh, yeah. Oh, sorry. I thought I was, okay, good, sorry. You still see my screen?
Yes, we can still see everything.
Yeah, okay, sorry. Okay, so when the levator veli palatini muscle contracts, it lifts the soft palate up to the back of the throat here. And that movement is required for speech and also in swallowing as well. So a very critical muscle is sort of a sling, so to speak. So if we look at, the problem is that, in a working functional muscle, we don't even have to think about this. But in particular, in some birth defects, including the cleft palate, this creates a huge issue for this whole structure. So if we look at the anatomy of a cleft palate, which can be combined with a cleft lip, so that would be an opening in the alveolar ridge or sort of the front of the mouth, versus a cleft palate is an opening in the palate itself. And so you can have a cleft lip and palate, or you can just have a cleft lip or a cleft palate. What we're going to talk about mostly is the cleft palate, because this is affecting this whole mechanism, in particular that levator veli palatini muscle. And so the cleft palate, cleft lip is repaired in relatively young infants, around 2 months of age-ish. Around 10 months of age is when the cleft palate is repaired. But it turns out after cleft palate repair surgery, only many children end up with what's called, they do not end up with adequate resonance or speech following the surgery, which takes a little while to develop because the surgery is done when they're infants, but it's still an important problem that hasn't been quite addressed. And so if you look at the anatomy of this structure, it kind of makes sense that it's still, it's not easy to reconstruct these muscles properly. So this is an illustration of how that levator veli palatini works on the left here. Is an animation made by Jamie Perry, where it shows you when it contracts, it's like lifting this whole levator, this velopharyngeal mechanism up. And so this is a kind of drawing here. So when it, oopsie daisies, when it contracts, so that's sort of this little lightning bolt here, it moves the soft palate up to the back of the throat, establishing this closure in the back of the throat. And so that's that closure that's required for healthy speech. And so, but many of the children don't end up with that ability to muscle to contract and bring the soft palate to back to the throat. You end up with what's called velopharyngeal dysfunction. So they're trying to use this muscle, but the soft palate can't get to the back, and you end up with air that goes between them. Here's an example of what that sounds like. So that's what's called hypernasal speech. As you can hear, sort of the tone of the speech sounds nasal, but then many consonants are very hard to say. For example, S requires you to be able to bring that soft palate to the back of the throat. If you can't, then you can't create the oral pressure to generate that sound. And pretty much all consonants, except for M and N, require that closure. So if we look at the BP closure itself, it has multiple kind of aspects. So one is this the velar component, this soft, this sort of tan structure, and then there's a pharyngeal component. Oops. And it turns out actually that if you look across people, it becomes even a little bit more complicated in the sense that, oops, I don't know what's going on there. When the soft palate moves up to the back of the throat, that's where kind of looking from the top here, you have the contraction here and the closure of that airway. It turns out some people are able to not only bring the soft palate to the back of the throat, but they can also constrict in the sideways direction. So we have, if you look across people, you have different, it's called closure patterns, sort of mechanisms by which they close this airway when they're speaking. And so it turns out that in terms of designing the appropriate surgery to address this velopharyngeal dysfunction, surgeons try to, and clinicians try to incorporate knowledge of how this closure happens into surgical or clinical management, but there's still a lot of questions because there's so much variability, as I was saying earlier, in the anatomy of individuals, that it's hard to understand, okay, which closure pattern are we seeing? And then also, which is the appropriate surgery to be done? And so that led us to thinking, well, we could use modeling to help because models are awesome. So this is a work done by Vi Tran in my, who just finished her PhD in my lab, who developed models essentially to uncover how the variability in the anatomy of the velopharyngeal mechanism influences the biomechanics of the soft palate. So we've been working in this area for a little while. We've generated some really nice 3D models, work done by Josh Inouye and Katie Pelland, both graduates from my lab, developed these nice 3D models to understand, first of all, just the mechanics of the velopharyngeal mechanism of many muscles that attach to the soft palate. So it's a complex geometry, but sort of limited population number because it was so intricate and took a long time to build these models. On the other hand, we developed some 2D models that were essentially a set of line segments and geometric assumptions about modeling the VP mechanism, this velopharyngeal mechanism, the levator and the soft palate. But so you can see here, it's quite simplified, but we did get some ability to then input more population variability. So what V set out to do is sort of merge those two concepts and develop 3D models of many subjects to determine how these anatomical variations affect VP function. So she starts with MRI data of the velopharyngeal anatomy. Interestingly, in the field of cleft palate, there's more and more proponents now of using MRI to guide clinical decision making, which is great. It's exciting to see that there's more and more value, because if you think about it, if you're going to, if you want to be able to address the variability in the population and use and develop a clinical strategy that is specific to the anatomy, you need to be able to measure the anatomy. And so MRI is really the only way, the best way to do that in 3D. So through collaborations with a number of folks, we've gotten a number of MRI data of the VP anatomy and developed 3D models. She has sort of a method to go from segmentations and multiple planes and using some CAD approaches to merge those to get models of the key structures. And so here's a kind of neat visualization of her model with the soft palate as well in the posterior pharyngeal wall. She did do some effort to streamline the CAD reconstruction process. As you all know, when you're developing 3D models from digitized data, there's a fair amount of noise. So she developed some ways to sort of facilitate smoothing and kind of mass production. So in terms of creating models of many people. And then message with tetrahedral elements, and we're using FE-Bio for simulations. The other thing that we have to do is assign the directions of fibers in the muscle. So this is this levator muscle in the purple we're looking at over here. The nice thing about that muscle is the muscle fibers are relatively simple. They sort of just go from end to end. So we section through. So we're doing half the muscle, assuming symmetry. And so the fibers use a flow simulation to find the fiber direction. Oops, daisies. So then material properties are assigned based on some published model and experimental data. We have a hyperelastic, nearly incompressible, transversely isotropic material to describe the muscle itself that includes the strain energy associated with the passive properties and the active properties. And then we have some other constraints and models of the other structures, the sort of the velum, in particular, the velum itself, the posterior wall here in purple. And then there's also another tendon that attaches into the soft palate that kind of constrains its motion that we modeled here. So in terms of the boundary conditions, we've sort of laid them out here. So really, it's an interesting mechanism. So it's the activation of this muscle that really drives all of the motion that's happening. So here's what a simulation might look like. If you've stimulated the muscle or activated it in the model, you've added that active strain energy, the muscle contracts and it pulls the soft palate to the back of the throat. And so one of the things that we've had to develop over the years for this line of work is figure out what are the output metrics of our models that are clinically meaningful, that actually relate to things that, let's say, a plastic surgeon or a speech language pathologist can understand. And so these are some of the outputs that we've arrived to. One is just the force that the levator applies to the posterior wall, essentially that reaction force between the two. Higher force means higher closure efficacy, like in terms of making a sealer. The other one is muscle activation required to achieve closure. So the idea being that if you require very little activation to get closure, that's sort of more advantageous. That means that it doesn't take as much muscle effort to speak. Or if you need a lot of activation, or you don't even, it's not even possible, that's suggestive, but more disadvantageous setting for the muscle. Then we also look at the amount of shortening, and that does kind of affect how much of that force length curve the muscle has to traverse. So so these are the metrics, and we also were interested in the anatomical, as I said, now looking at, for across the models, looking at the anatomy, things that you could measure directly off the MRI and how that influences the functional predictions from the model. And these are really inspired by what the field does in terms of MRI studies of the VP mechanism. So we had a kind of, we call them family of models. These are different models that we created and looked at, we wanted to calibrate them with respect to data that existed in the literature. So one is there had been some nice studies, EMG, where they measured the EMG of the muscle, elevator muscle, and looked and measured closure force using a kind of a bulb that was placed in the back of the throat, and looked at how they related. So more activation, more closure. And so we picked one set of measurement or sort of parameters and used that for all the models so that we could really look at the effects of geometry. But overall, they sort of, in general, match the trends of the experimental data. And then we also had dynamic MRI, which is relatively low resolution. We're kind of looking at a dynamic MRI of sustained phonation in speech. And we looked at how the model predicted, so the motion of that soft palate as compared to the dynamic MRI, and used dissimilarity coefficient to compare model versus dynamic MRI, and saw really high relationship in terms of coefficient, and even contextualize that with comparing the dynamic MRI at different other time points. So this was the maximum closure, and we said, okay, well, the model must do better than any other measurement at any other time point, and indeed it does. So now to the model analysis. If you look at across the models that we have, we looked at things like the levator. This is levator velli palatini cross-sectional area. So bigger muscle means stronger muscle. And that is predictive of that force of closure at, we did specifically at 50% activation. So that makes sense. Bigger muscles generate more closure force in that closure. And then sort of the opposite holds true. It's not as tight of a correlation, but in terms of the muscle activation required for closure, the stronger muscles, the bigger muscles didn't require as much activation to get to that full closure. Again, makes sense. But then we looked at other things like the velar thickness and as it relates to these metrics, and then the velar length, and those also have a relationship with that closure force. So illustrating this impacts to some degree some of these surgical approaches that are done because, for example, this Z-plasty technique, one of the techniques for cleft palate repair, and even revision surgery is intended to lengthen the velar, the velum. So if you're dealing with a subject that has a pretty short velum, this does indicate, this type of surgery would be beneficial. This result does sort of highlight that. And then, we can have a similar story for a different approach that's used to thicken up the levator. But the other interesting thing that we found is that although, I talked about these two different closure patterns across different people, and we did find significant differences in some of the functional and anatomical features between these different kind of closure pattern subject, and that these closure patterns were essentially assigned by a trained speech language pathologist using a nasendoscopy procedure to kind of visualize that motion. So clinical assessment. So we did see some differences, but we also saw that there's a bit of a trend though, in terms of that levator function. It's not like one or the other really, which I think is an important thing to think about in terms of looking at across population, that people aren't just one or the other. There is kind of a continuum. So these are just some ideas I'm happy to chat more about where we're going with this work and some of the other implications, but some thoughts about integrating finite element modeling, imaging, and clinical assessments to make some insights about anatomical variability. The next application I wanted to explore, kind of shifting gears a little bit, is another different muscle, in particular breathing, and as it relates to muscular dystrophy. So just a little bit of background on muscular dystrophy. It's a fatal muscle wasting disease that has these devastating impacts across a range of scales. So it's a lack of a functional dystrophin protein at the muscle cell membrane. So there's this key protein called dystrophin that connects the muscle contractile apparatus through the cell membrane to the ECM. So it's the lack of that protein that's the kind of genetic basis for the disease. But what we see over time, that we see changes in the composition of muscle. So a young muscle in a child with Duchenne muscular dystrophy, their muscle will look relatively healthy, but over time, because the dystrophin lacks, you'll end up with damage to the muscle, fat infiltration, fibrosis, things like that. And you also ultimately see a loss in muscle strength over time. And so you'll see lower limb muscle degeneration, but you also see respiratory failure, which is ultimately the cause of death. And fortunately, there's been some innovation in terms of managing the respiratory failure, i.e. the essential loss of the respiratory muscles. So we have more assistive devices to help with breathing in these individuals. However, there's still a lifespan is roughly 30 plus or minus a little bit. So there's a long way to go in terms of understanding the mechanism of this disease and how to combat it or develop ways to address the respiratory insufficiency. So the diaphragm muscle is essential for respiration. So that's the muscle we're focusing on here. So when you inspire, the ribs move up and out and the diaphragm flattens, so it contracts. And then when you expire, the diaphragm relaxes and the ribs move down and inward. So this is what it looks like if you track the sort of top of the diaphragm using MRI and inspiration, expiration in a 9-year-old versus a 26-year-old boy with DMD. You see a great decline. By their late 20s, their diaphragm just doesn't move much anymore, which sort of speaks to this respiratory insufficiency. So we're interested in really what's leading to that, and what are the structural changes and variations in that diaphragm muscle tissue that leads to this loss of respiratory function. So we've focused on the mouse model for the disease for multiple reasons, because we can really interrogate the muscle experimentally, but also the MDX mouse model does replicate the diaphragm dysfunction seen in DMD patients. So you can see a reduction in strength and elasticity and increases in collagen density. So that's speaking to that fibrosis. Interestingly, though, in the field, we've seen that that if you look at the collagen amount within the diaphragm muscle, the area fraction of collagen, you don't see a relationship with the stiffness of the tissue. And so this has been kind of replicated a few times. So it's not a simple story like, okay, there's fibrosis, so it's stiffer, it doesn't move. Like it's not a simple story like that. So we've been conducting experiments to characterize the mechanics and function of the diaphragm. So looking at, so Hunter Wallace just finished his PhD in my lab also. He did some sonomicrometry experiments looking at the in vivo strains of the diaphragm in both healthy and dystrophic mice, and then also did some biaxial mechanical testing to look at the ex vivo tissue properties. And so just looking at some of these, biaxial tests that he did, looking at both wild type and MDX, and we're looking at different ages, partly to kind of examine the progression of disease using this biaxial cell scale system you can see sort of it's a biaxial testing setup. And so as he was doing these experiments, we really wanted to look at the contribution of collagen. So he digested out the collagen. So did the test before and after digesting the collagen with the Liberase solution. And also used the biaxial system to essentially characterize the stress strain curves in both the along fiber and cross fiber direction. In the system, we can also track the deformation or the displacements of specific regions on the tissue and use that to find deformation gradients. And then, of course, we're using the reaction forces and those deformation gradients to calculate stress. So we also wanted to quantify the amount of collagen using Pico series red imaging, where we looked at the collagen amount and distribution within the samples. So after we tested them, we would section them and look at things like the amount of collagen. We looked at, we're interested in both the epimesium and intramuscular ECM. So in his experiments, he looked at the stiffness in the along fiber direction. So the wild type are relatively compliant, and then the MDX, and this is before anything's been digested or anything, are much stiffer. But then, of course, when you digest the collagen, the stiffness goes down significantly. The wild type kind of almost goes to zero, but in the MDX, it's not quite as profound of an impact. Interesting, the cross fiber direction is also interesting to think about, because in sort of regular skeletal muscle, we think about a lot of the properties really being along the muscle fibers. So, the stiffness really is, along muscle fibers, and the cross muscle fiber direction is usually not as kind of, studied. But what we see here is that the cross fiber direction is actually, in the wild type, for example, is actually the stiffness is higher than the along fiber direction. And then also we see a significant cross fiber direction properties in the MDX as well. And then we see how the digestion, similar to the along fiber, does influence both of those, though, in the MDX, you're still left with a little bit of stiffness there. When we kind of quantify the collagen area fraction with the histology, we see that the 6-month-old mice have more collagen than their age-matched controls. So the 12-month wasn't nearly as much in terms of area fraction. And then we see post-digestion, we do see that that since we wanted to use the same digestion protocol, there was more change in the there's less collagen area fraction in the digested wild type than the MDX, because they just started with more, but there is a decrease in that properties. Interestingly, though, kind of back to that, what I was pointing out at first, we did also find that the collagen area fraction, if we looked across all the samples, there's a lot of variability in the area fraction and the stiffness, but there, it's only a very loose correlation between area fraction and stiffness in both directions, which really speaks to this idea that it's not just area fraction of collagen that influences the properties of a given tissue sample. So we also wanted to look at the in vivo strains as well, because that was sort of taking the muscle tissue out and just putting it in the tester ex vivo. Let's see what happens in vivo. And so here we're looking at, I think this is an animate, yeah, I think it was animating, but we measure these sonomicrometry crystals. You're able to track the relative motion of these crystals and look at the displacement over time. So we can look at the along fiber distance, so between the two crystals that are positioned along the muscle fiber, and then cross fiber. And so as we see during inspiration, is this like little blip here, that the along fiber contracts as we'd expect, and then the cross fiber actually moves apart. And so the strains are calculated between the breath and resting length. So this kind of in vivo strain was between this sort of resting length and then the contracted length. So the 6-month wild type long fiber strains were greater than the age-matched controls. That's interesting to note, that the 6-month wild type did shorten more in vivo than the 6-month controls, which is interesting. I think sort of in consistent with the limited muscle or diaphragm excursion. And then same thing goes for the cross fiber strain. So there does seem to be a relationship between the long and cross fiber behavior. More long fiber strain seem to equate to more cross fiber strain. And then if we looked at the collagen area fraction, again, there was an interesting but weak correlation between the area fraction and the in vivo strain. So we already saw that it was a weak relationship with the ex vivo stiffness, and now another weak relationship with the in vivo strain, though kind of a trend. But what we realized, though, is that we probably need to start thinking about collagen more than just area fraction, sort of diving into the details. And so that's led to us considering the organization of the collagen, and started to use different imaging techniques to characterize the arrangement and orientation of the collagen fibers. And so that's where Riddhi Sahani, who's currently a PhD student in my lab, has been working, looking at samples where she actually digests the muscle fibers out, so she can specifically look at the organization of the ECM. And we've been focusing first on the collagen, given the collagen makes up a lot of the ECM. So there are a lot of ECM elements, but we focused on the collagen to look at its organization. And so using SEM imaging, where we see the collagen structure kind of beautifully illustrated here. And so where she's looking at the relationship between the muscle collagen fiber and actually the muscle fiber. So she takes great care to quantify or understand what is the fiber direction relative to the collagen direction. So one thing that we see very consistently is that the collagen, so if these images are arranged such that the muscle fiber direction is horizontal. And if you see the collagen structure here, it's vertical. So it's essentially perpendicular to the muscle fiber direction, which that was actually quite surprising to us. And we see that consistently. But then she also looked at other measurements like the straightness parameter. She measured, which essentially she looked at the relationship between the length of the collagen and the kind of Euclidean distance. And looked at how that varied. And then she also had developed this image analysis framework to look at the local collagen direction using the radon transform. Essentially, by binarizing the image and using the radon transform, she could find the local collagen direction for a window, sort of a region of the ECM, of the SEM image. So here's some examples of what she found. So here's a 3-month wild type versus a 3-month MDX. And you can see from the image that it's a lot wavier in the 3-month wild type than the MDX. And so this is what her algorithm would give you, is this variation in the orientation angle of those fibers relative to the muscle fiber direction. And you can see that here versus the MDX is a lot more aligned. And we see that trend in the MDX, especially at the 3-month, a very big difference in that collagen alignment, which sort of varies over the ages. So interestingly, she also from that radon transform could look at the collagen pixel ratio and also the direction relative to the fiber direction. The collagen pixel ratio is relatively preserved, not a lot of variation. And there was really no difference in the orientation of the fibers relative to the muscle fiber direction. So those weren't really different. But then once she was looking at the straightness and the alignment of the collagen fibers, that's where we saw a big difference in terms of the MDX were more straight and then also more aligned in a particular direction as compared to the wild type. So back to this sort of question of the relationship between structure and function and the variability, she decided to create some models to understand how this variability in structure influenced the mechanical properties of the the structure. In particular, this is the epimysial layer of connective tissue in the diaphragm muscle. She used her radon transform, an algorithm that characterized the direction, and inputted that into a finite element model where each element was essentially a pixel from this analysis. Where she modeled the ECM as a coupled solid mixture, where she had tolinear collagen fibers that she defined, and then she had a ground matrix. And then she used her radon transfer to find the material axis for each element. Essentially, what is that fiber direction for each element? And she simulated an equal biaxial stretch. So here's some of her results, her finite element model results in the cross muscle fiber and along muscle fiber directions between the different cases. And you can start to see this is the visualizing the element stress. And so we saw that the along muscle fiber stiffness increased in the wild type, while the cross muscle fiber increased in the MDX. So this really shows you how, like, not only that sort of this rearrangement here led to sort of changes in the biaxial properties in a maybe not perfectly intuitive way, where the MDX, now the along muscle fiber stiffness, was actually less, but the cross muscle fiber stiffness was more. And then she looked at the straightness and collagen fiber, and those were real predictors of the SEM-based image model. So looking at over a range, so the collagen fiber direction versus these stiffness values didn't have much of a relationship. But then once you looked at straightness or strength of alignment, those had a really great predictive capacity. So really highlighting the point about the organization of the muscle, of the collagen, and how that influences the mechanics. So just this last tidbit, I wanted to share, sort of zooming out, sort of branching off the muscular dystrophy application. I wanted to share some sort of into the clinic concepts. So, with our modeling, we do these very detailed models, the MRI based, with the meshes and all this interesting constitutive models and all those things. I think in terms of the concepts that we're doing, it's nice to also think about how can we make these concepts clinically useful, and how can we leverage the knowledge from these more detailed analyses to things that can be in the clinic. And so that's been a project that we've been doing in collaboration with the Muscular Dystrophy Clinic at UVA, looking at imaging biomechanics assessments of muscle in both Duchenne muscular dystrophy, which I talked about, DMD, but then also spinal muscular atrophy, which is a severe muscle atrophy due to the death of a motor nerve. Really inspired by the fact that clinical assessments of muscle function are subjective, and they really lack sensitivity, in particular when these children have severe muscle dysfunction. You really have to rethink about how you're assessing muscle so that you're addressing, you can detect fine changes, because there's a lot of now clinical trials to try to develop drugs to help combat these diseases. But, these trials really rely on good measurements of changes. And, as they get better, you're going to see smaller improvements over time, and then which hopefully will lead to bigger improvements, but we need a way to measure it. So Ali McGrady has been developing a technique to do ultrasound measurements in SMA, DMD, and typically developing control. And she's developed a relatively good sort of quick way to take these children. They don't have to be able to take too many directions or anything. And she can very quickly image with ultrasound their muscles, their upper extremity muscles. She's looking at a few upper extremity muscles, including the biceps and the brachialis, and looking at the cross-sectional area and the echogenicity, which the cross-sectional area, as we know, relates to force generating capacity. I was already talking about on one of the other muscles. But then echogenicity is thought to be relating to the amount of fibrosis and fat in the muscle, which we would think about that relating to the force per unit area that a muscle could generate. So she's been measuring these features using both longitudinal and transverse ultrasound, and developing some relatively simple models to address these changes. So looking at cross-sectional area, echogenicity, and even looking at how estimated specific torque. So she takes a measurement. We're taking also dynamometry measurements of strength and looking at how that relates to the CSA and the echogenicity. And so these are kind of examples of what we see in these three cases. Some in the healthy versus the DMD, what we see at the younger ages, it looks a little healthier, but as you see at the older ages, it's much brighter, which is suggestive of this of the fat infiltration and the fibrosis. But in the SMA, while in the DMD you see bigger muscles that are bright, you see in the SMA you see more atrophied muscles that are also bright. So she's been looking at how these normalized CSA compares in the groups, and then also the echogenicity does seem to kind of relay, like the healthy values are lower as compared to the others. And then lastly, now looking at the relationship with muscle function, as I was kind of talking about before, looking at specific torque. So that was taking the measured torque and dividing by the CSA and normalizing for differences in body dimensions. She could see a really strong relationship with that echogenicity, showing this a relatively simple measure that she could use to understand the functional capacity of the muscle. So she's now using this result to kind of develop some muscle scoring just based on CSA and echogenicity, and see if she can use that to track longitudinally changes in the muscle over time with different drug treatments. So just to finish off with a few new lab directions, kind of building off this variability, we have a new NIH project that's sort of just getting underway, where we're looking at now kind of within healthy individuals, examining sex differences in scaling and function. So doing a more thorough job of addressing our study population in a way that we could look at variability between sexes and then within sexes in terms of scaling of muscles and strength. And then we're also looking at modeling estrogen-specific behaviors. I didn't talk today about some of the work we're doing in collaboration with Shane, and this is also what Katherine did when she was here, at agent-based modeling of muscle regeneration. We've been doing that a lot, coupling with mechanical models. We're kind of moving into understanding how sex hormones influence the regeneration. So with that, I want to thank students and collaborators and funding sources, and for your time. I apologize for the Zoom difficulties in the internet, but thanks a lot.
Hi, Sylvia, thank you so much for a really great presentation. Sorry about the technical difficulties, but I think it worked out in the end with you able to share. Now I want to open the floor to any questions.
Hi. First, I would like to say the project sounds really cool. I'm personally very interested in gender difference. I'll take a look. But the question I have is more the curiosity. When you showed the palate muscle, I was wondering, what do you think when people have different native languages? Do you think there is some kind of a different structure there going on? Like, for example, I noted my mother tongue is Portuguese, and some Chinese people were with me learning Finnish, and for whatever reason, they could never do this r sound.
Yeah.
So I started to wonder, do you think the structure is very significant related to your mother tongue, or is just, you try hard and hard and hard, and then you get there?
It's a good question. There are some studies that show there are racial differences in some of these craniofacial and velopharyngeal anatomy anatomical parameters. So there could be some structural sort of aspects. I think there is definitely a motor control aspect too, and maybe related to motor learning at an early age with some of these specific sounds. What's interesting about the hypernasality is it's an issue across all languages. So it's a very sort of overall uniform issue, regardless of of language, but I think there probably is something there in terms of structure. And whether that's nature or nurture, that's an interesting question. But yeah, for sure, I think that's be an interesting thing to explore also.
I agree. Thank you.
Any other questions from the audience?
Well, I have one. So thank you very much, Silvia, for the great talk. It's a curiosity. It's more technical questions, I would say. When you're assessing the difference of stiffness of the fibers through the biaxial test, so you're choosing a strain of 8%.
Yeah.
Any specific reason for this choice?
It's a good question. Yes. Yes. One of those decisions that you go back and forth with the student on and much belabor it a lot. We actually did in his thesis, we looked at 4% also. We were thinking there might be different contributions at the lower strains and more of a toe region versus the 8%. So it's similar. The kind of bottom line is that you get about the same relationship. So that's why I only showed one. I mean, ultimately for a model, you want the whole curve. And I think that's actually one thing that he's still working on his paper to describe this. And one of the challenges for us in biomechanics is like when people present these stiffness values, they have to be at a specific, for all these tissues, they're nonlinear. So you got to contextualize it with the strain or, but a lot of people don't. They might even give it a different strain values. But yeah, I think we really, I presented that really because the answer in terms of the relationships and differences between the groups didn't depend on that, where on the curve we were looking. But for sure, I think for a model, you'd want sort of the whole thing.
Yeah, thanks. And another question I have is that, so you're looking basically at the passive behavior of a dead tissue when you're doing these tests. So, but I'm wondering, so what is actually the interplay between the active part and the passive part when this protein pleiotrophin, if I remember correctly, is affected? So does it impose, actually the change of stiffness of the passive part, does it impose a particular burden on the active part, and which then would explain the fate of the muscle along the years?
Yeah, no, it is thought like one of the functions of this protein is to protect the membrane from stress and active contraction. That's thought to be the mechanism by which the muscle, or one of the many mechanisms by which the muscle degenerates over time, is that regular active stresses, which wouldn't damage a healthy muscle, damage the dystrophic muscle. And so we've actually done some modeling of that and looking at with and without dystrophin how that leads to stress and strain in the membrane and also the ECM elements. So yeah, there's definitely an interplay there. And yeah, with our passive experiments, those are really kind of focused in on those passive mechanics. But I agree in terms of like one thing that Hunter, the student who did these biaxial tests, his vision, and he's sort of he made some good progress, is to integrate the passive mechanics and the sonar data into a 3D FE model of the diaphragm that also has active properties that he could simulate, and even simulate the changes in those, and see how that interplay between the passive and the active. Because the other piece that's useful is thinking about the treatments for this disease. Like there's a lot of effort on essentially replacing the dystrophin protein, which would hopefully kind of sort of make these active, this sort of damage mechanism, stop, but then also address the active, that the muscle fibers remain healthy and active. But then there's other diseases that are more, or treatments that are more focused on the fibrosis and the development of fibrosis, sort of addressing the passive part. So that's why in the diaphragm in particular, because it seems like these passive mechanics actually play a big role in the function of the muscle. It'd be interesting to know, like, do you need to be replacing the dystrophin protein to have the muscle work, or if you have relatively weak fibers but with passive mechanics that are more kind of conducive to motion, might you be able to preserve that respiration longer? So those are the things that we've been thinking about for this model.
Switch off now. Yeah, thank you very much. Yeah, because I was particularly struck by the fact that the transversal stiffness is actually affected differently. So when you have the active part coming on, actually also Poisson's effect on muscle transversal deformations are particularly affected. So, and then my second question was whether it can have applications to support regenerative strategies, but you've just answered that. So,
yeah, good. We're on the same page.
Okay, and then I also actually had a question about the ultrasound work that you presented at the end for spinal muscular atrophy and DMD, and how you had found that the echogenicity really correlated well with the torque measurements. Have clinicians been able to use those measurements to then kind of like understand like where they're at within the disease and prescribe certain like physical therapy or other regimens, or do you see that happening in the future?
I hope so. Yeah. We really hope so. That's kind of where we're going. As you know, Rebecca Scharf, she's excited. She and Ali went to the Muscular Dystrophy Association meeting earlier this year and got some really good interest, especially from the pharmaceutical companies that are running clinical trials, because this is a good way for them to see if their treatment is helping. And one of the cool things about this particular case is that if you don't, so we're doing the torque measurements on these kids, sort of inclusion criteria for our study is to be able to do them, but a lot of these kids have cognitive defects to the point where they wouldn't be able to do that task well enough. So right now, all those kids have to be excluded from clinical trials because they can't do what the drug companies need them to do to assess changes. But if we can essentially show that through a combination of ultrasound measurements, we could replace that, then that would allow just much more inclusive approaches to addressing changes and sort of make it easier for people to do these types of things on all the kids, not just the ones that are specifically capable to do all these specific tasks. So that's an exciting kind of avenue that Ali's been exploring a little bit more.
Yeah, that's super exciting. That would be really incredible if they could use it like that. Okay, so I think that's it for questions. Thank you so much for joining, Silvia. I know it's really early in your time as well, back in the US. But yes, thank you so much for a really great talk.
Thank you. Thanks so much for having me.